Hi, Rory. You can turn on your microphone.
Hi.
Hi, can you hear me?
Yeah.
Okay. So just to make sure with you, I want to know who here speaks Hebrew and who speaks English, so I know how to run the course. So you only speak English, or?
Yes, I speak only English. Sorry.
Okay. Oh no, that's fine. That's fine. Then we'll do the entire course will be in English, as long as there's
me
one.
that I sent you the mail. Thank you really much.
Sure, that's fine. It's no problem.
It's me that I. Okay, thank you really much.
Okay. So where are you from? Whose lab?
Okay, I'm from Italy, and I'm in the lab of Professor Ayelet David.
Okay, great. Hi, Nadav.
מה שלומך? מה הולך?
על הכיפק. תכף נשמע קצת עליכם, אבל מאיזה מעבדה אתה? של מי?
מהמעבדה של פרופסור אוהד בירק.
בירק, נהדר. אוקיי. עובד שם הרבה עם יובל?
לפעמים.
עם סולודש. היי עמית.
שלום, רגע אני אסדר פה את העניינים. אהלן.
מה העניינים? תגידי לי ככה בגדול מאיפה את? עוד לא התחלנו, זה רק... לא בארץ. אני מנסה לקבל קצת אינדיקציה. נעשה את זה רשמי עוד מעט.
אני עם נדב גם מהמעבדה של פרופסור אדביר.
וואו, יש לנו פה כוח
יש עוד
מנצח.
נציג גם.
יש עוד נציג אפילו? וואו.
יש עוד 2.
נהדר.
אני גם כאן.
אופק, אוקיי.
סליחה שאני בלי מצלמה, אני פשוט אוסף את הכלב שלי עכשיו מהווטרינר, ואז אני אגיע ואפתח מצלמה.
בסדר, נראה מי יגיע קודם, אתה או הכלב. איזה פסיכדלי אתה היום. היי נטלי.
היי, שלום.
אוקיי, אני רק אגיד את הכמה מילים בעברית, אבל אנחנו נהיה חייבים לעבור לאנגלית, כי יש לנו פה סטודנטית שלא מדברת עברית. So I'll now move to English so that Rory, can you hear us?
Yeah, I can hear
Okay.
you.
Okay, great. So we have Rory with us. I think we can wait a few more minutes before we start. So we will do it, and all those of you who prefer Hebrew, you have Rory to blame. But I'm just joking. So we will do the entire course and lessons and discussions in English. But yeah, I don't think that anybody here would have a problem with that. It's not going to be too complicated to none of us, I hope. And if there is someone here that the English is a little bit of a problem for him or her, then let me know, because it would be preferable to do everything in English, to write the work in English and the discussions in English. But again, it's not necessary. It's not an obligation. So if any one of you has a problem with that, just let me know, but I don't think there should be a problem, okay? So we'll do it in English. So as I said, we're gonna wait. I think that now we have, how many? 2, 4, 7 people here. And there are a few more people that are registered to the course. So maybe I'll wait till 2:20 or so and let everybody connect. I'll just let you know that we are going to, I'm going to start by describing the course and the prerequisites and the obligations that you have in this course, which is not too much. Here, we have Matan here as well. Hi, Matan. We're not contagious on Zoom, so it's fine for you to take off your mask. מנסה להבין לאן הוא הגיע. מתן, אתה שומע אותי?
אנחנו, נדב, מתן ועמית יחד באותה מעבדה, ובגלל זה היום אנחנו עם מסכות, אבל הנה מתן.
כן. שומעים אותי עכשיו? מגניב.
אני רואה אותו שם מאחור. Yeah, I can see him behind נדב. אוקיי.
הנה נדב, הנה עמית.
Wonderful. So, you know, I'm thinking maybe we should all come to your lab. It would be easier for everybody, I think. Yeah. Okay. let's wait till 2:20 and then.
It's a good idea. We love to have you.
Oh, good. Just have some refreshments ready, something to drink. Either. Okay, so 2 more minutes and then I'll start. I don't want anybody who comes late to miss anything. Noah, can you turn on your camera? תוכל להדליק את המצלמה?
כן, אני מדליקה רגע.
היי, יופי.
סבבה?
היי, מי הצטרפה אלינו עכשיו? את יכולה להדליק את המיקרופון? היי ביאן,
היי,
מה נשמע?
בסדר.
Okay. So we're going to start in 1 minute, okay? We were waiting just for you. חיכינו רק לך.
סליחה, תעקבתי קצת.
הכל בסדר. Okay, so I think we can start. So hi everybody, first of all, and welcome to the Metabolic Communication course for grad students. So we have, I think we have different levels of students here. So maybe we'll do a small, you see we're not too many people, there are very few here. I just want everybody, all of you, you know, we'll go one by one, nothing too complicated, okay? Just say so that we can meet you, because we're going to be a small group and we're going to be doing many discussions together the entire time. You'll get to know each other very well, but just tell us just a few details about yourself, nothing since birth, just, you know, which lab you're in, what is your degree, okay, are you a master's or a PhD student? You can also tell us what you're studying, because this is going to be something very pivotal in this course. Okay, it's going to be very important. Unlike other courses, I think, I will try to put your projects in the center. Okay, it's going to sound weird because maybe most of you or all of you are not involved in metabolic communication, but I think that by combining the knowledge that you'll have here with your project, that's going to be something very special to do. And so I'll let you know how we're going to do this later, but so let's do a small cycle of self-presentation. Okay, so Nadav, do you want to start? You look like a person that wants to start.
You're right. I'm Nadav from Professor Rad Birk's lab, and I'm an MD PhD student and studies the molecular basis of monogenic diseases.
Okay, great, thanks. Ruri?
Hi, I'm Ruri, and I'm a PhD student in Mr. David lab, and now I'm doing a research about cancer and drug delivery.
Thanks. I'll just write this down because it's going to be important in this course, as I said. Yeah, Natalie?
Hi all, I'm a PhD student in the lab of Professor Asaf Rudir, with also under the supervision of Isana Wetzler from Andasat Marachot Meida, which I'm not sure how to translate, sorry. My PhD is...
That's fine.
Connection issues. My PhD is about finding a microRNA biomarker that will reflect the health of the adipose tissue in obesity.
Nice. Relevant. Yeah.
Yep.
Okay. Aaron.
Hello, I'm Aaron. I'm a PhD student. I work in Eli Lewis lab, Professor Eli Lewis. My PhD will be in tissue repair. Now it's in the beginning.
Achla. Great. Thanks. תודה, אהרון. Yeah. Matan.
Hey everyone, my name is Matan. I'm from Oad Birks Lab. I'm an MD PhD student with Nadav and Ofek, and Amit is with us in the lab. And we studied the molecular basis of monogenic diseases.
Great. So we know exactly what Ofek is going to say.
Oh, do you?
Yeah, I'm Ofek. I'm from Professor Ohad Birks lab, and I study monogenic diseases.
Yeah, something like that. We studied the genetic of rare or unknown diseases, and I think my main subject is the conduction in the heart or brain and the eyes, diseases of the eyes.
Okay, great. Thanks.
And
Don't,
my name is
what?
Just saying, and my name is Ofek. I think you said it already.
That's important. Yeah, that's important. Just make sure you are focusing on the road and not the phone.
No, I parked. It's okay. I'm at the vet place.
Okay. But make sure you're parking in the right place. We're not refunding you for any tickets you get.
That's important.
Okay. Thanks, Ofek. Noah? By the way, if you prefer, anybody here who prefers to talk in Hebrew, that is fine. I will translate you. Just feel free to do whatever is best for you.
No,
Mute. אתה על מיוט.
סליחה. אני נועה, אני סטודנטית למאסטר במעבדה של אודי, דוקטור אודי אוחנה. זהו, מה, לפרט על הפרויקט שלי?
לא חייבים. זה בסדר, okay? So Noah is from my lab. In case you didn't know, it says Ehud, but you may very well call me Udi. I think it's going to be even better. And Noah is actually studying the metabolic effects of metabolite transport, or the physiological effects of metabolite transport in metabolic communication processes, and specifically in cancer cells. Okay, I think that we are now with Bayan and Amit. Amit, do you want to go first, and then Bayan will be the last but not least.
Okay, so I'm Amit. I'm also from Birk Lab. I am a master student, and one of my projects are protein and the connection protein, and that's it.
Okay, the connection was very bad. I couldn't hear anything. Was it just me?
לא שומעים אותי? אז
I think Nadav is drinking all your Wi-Fi.
Now it's better?
Okay, great. Put masks on.
Okay. So my name is Amit. I'm also from Oad Birke Lab. One of my projects connect to vinculin protein and the connection with actin. And that's it.
Okay, toda. And last but not least, Bayan. Yeah.
Yes. Hi, I'm Bayan. My bachelor degree was in pharmacy. I'm doing my master degree in Galile's Agam Lab. And we are working on the effect, if global warming has an effect on bipolar disorder. We are searching after if there's any changes in biochemical factors or in behavioral, if there's a behavioral effect. That's
And is there? Is there an effect?
yes.
There is. Okay, that's surprising.
Yeah.
Okay, wonderful. Okay, so thank you, everybody. Now that I know a little bit more about what you guys are doing, it's not that I'm going to remember anything. So the first thing is that I'm going to ask you where we're going to have, maybe I'm going to open and put the link to a Google Sheets site, and I'm going to put it in the Moodle. And it would be nice if next to your names in this sheet, you'll be able to write your projects. Okay, and soon I will, well, I started telling you, but I'll go deeper into it and explain what we're going to do in this course and why your projects are very important. Okay, in general, what we're going to do, you will have the program in the website. I think you already have a link to a website that we're going to use. Too many websites, but again, the Moodle is not very user-friendly in this matter. So Eli Luis, who is going to run this course together with me and maybe often be here. He is here actually listening. And he made this, he built this site, and you'll be able to have all the information of this course, the movies and everything is going to be there. You can see actually the program. It's still not detailed, but in general, what we're going to do in the first two classes, which means today and next week, you are going to be rather passive, okay? Passive, it means relatively, because I do want to have a fruitful and meaningful discussion with you today also, but I'm going to do an overview of two classic I would say disciplines that are relevant to this course. The first is metabolism, and we're going to do this just to adjust, okay, to start from the same point, because I know that each and every one of you comes from different disciplines. You probably don't remember all the material that you studied in your first degree and metabolism and things like that. So we're going to first talk about those two major disciplines, which is first, a few classical metabolic processes. And then I'm going to also talk about intercellular and inter-organ communication, okay? Because what this novel field of metabolic communication, and more specifically, it involves the microbiome and other fields like immunometabolism that you may or may not have heard of. It actually combines all those basic disciplines. So we have to start with something classical, and then we will move on. But after those two meetings, and actually if I'll be able to finish everything today, then maybe we will start our discussions and sort of journal clubs from next lesson already. So basically you'll have a paper for each week, okay? And you don't have to read the entire paper, the entire manuscript. Each manuscript will deal with different aspects of metabolic communication. You can see it on the list in the website, and you will have to read a part. Okay, I will assign different parts of this manuscript to each and every one of you, and then when we go back and when we meet in this, can you hear me? Because it says that the internet connection is unstable. It really is unstable, I guess. Can you hear?
I can hear fine.
Yeah, there is some kind of problem. Don't know why. Let me try to connect to the line itself. Yeah, I can see that. Are you back? Can you hear me now?
Yes.
Okay, when did you lose me? Probably at the very beginning when I started the monologue. Okay, I'll just go back a little bit and tell you that in the coming lessons, what we're going to do is that I will have a manuscript for each and every meeting, and we will divide the manuscript to different parts. I think that currently we are, let's see, how many are we here? I think 8 or 9, right? Or more? Amit, you're here twice, right? Okay, so I think we have 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, there are 9 people here, so we can divide to either a 5 and 4 group or smaller groups of 3, we'll think. We'll see how it's going to be best. I like the 5 and 4 better. So we'll have 2 groups, and each person in this group will be assigned part of the manuscript. Okay, and we will start by, and again, for each and every meeting, I will choose one person, and his project is going to be the highlight of the meeting. And of course, this is why I'm asking for your project, for the topics of your project, so that I can assign relevant manuscripts to your projects, okay? And then the aim of each meeting will be first to understand the holistic view, I would say, of the manuscript that you will discuss, and understand the manuscript and the topic that the manuscript deals with. And the second aim would be to advise to the person whose topic is in the center of the meeting, of the class, on how to apply the specific area that will be discussed in the paper to his project, okay? Because the final assignment in the course, and that's a personal assignment that each and every one of you would have to submit, and that's a project in which you describe how you basically use the knowledge in this that will be provided in this course to implement or to combine in your project. It doesn't mean that you have to do it, of course, okay? But you will have to design a project on how you implement metabolic communication into your work, into your PhD or master's project. Okay, is that generally understood? For those of you who are not frozen, it's very hard for me to say who's frozen and who's just sleeping. Okay, great. Any questions? No. Okay, so we can basically start. I'm going to share my screen with you.
I have a question.
Oh, great. I really like questions.
No, I just wanted to ask about the grade. If there's an
Yeah.
exam or the...
No, yeah, that's exactly what I said. There is no exam, only a project that you have to submit, okay? It means that you're going to write a work of, I don't know, that spans 4 or 5 pages, nothing more than that. Something very concise where you will have to show how you implement metabolic communication, a specific aspect of metabolic communication into your own real project. It means that if you are working on the effects of global warming on mental disease or anything like that, okay, I just made it up. I know it sounds different, but I just made it up. So how you can use a specific aspect of metabolic communication in your own work, in your own project, okay? And later we're going to say exactly how the work is supposed to be formatted and which specific things we want to have in it and how you design specific experiments. But again, it doesn't mean you have to do that, okay? It's just sort of a proposal, I would say.
like a research.
בסדר, ביאן?
It's like a research proposal.
Yeah, exactly, like a research proposal. Exactly. Okay, great. Okay, so let's talk a little bit. Actually, it's not going to be a little bit. I hope to finish earlier today than 3 hours because it's pretty massive, but you'll see that when we divide next week or maybe the week after to discussion groups, it's going to be much more fun and the time is going to pass much faster. Okay, so let's start a little bit with metabolism. Okay, before I start, I just want to ask who of you did an introduction metabolism or biochemistry course? I think I presume everybody here, right? Yeah, I'm sure that the MD students are past that. Aaron, I know you did. Noah, obviously. Amit, where did you do your first degree?
In biology here.
In biology.
Ben Gurion.
Okay. So Natalie?
Yeah, I did a medical laboratory, but a while ago.
Okay.
Kind of
that's fine.
Maybe.
That's why we are doing this now. That's fine. Okay. So you all have reasonable to good knowledge in metabolism. Okay. So basically metabolism, or I'm starting with this because I'm sure some of you ask, or maybe some of you know, what is the difference between metabolism and metabolic communication? Do you have any idea? And if those of you prefer to talk in Hebrew, that's fine. I can translate. Don't feel bad. What do you think is the difference between metabolism and metabolic communications? We both have the metabo word in them, but what is the difference? So basically, it's very hard to tell. I understand why you guys are being quiet because it's not that obvious. Metabolic communication shouldn't basically be different than metabolism. So the question is, why is it suddenly becoming a very hot and timely topic that everybody is talking about? While metabolism is, kind of anachronistic, something older that you find in textbooks. And I think that it's just a matter of fashion, okay? Basically, I think that the reason that we have metabolic communication as a novel field is because of all the high throughput methods that popped. And now we can do not only metabolism and look at the classical enzymatic function, we can actually say that we can look at the entire metabolome. Okay, so the guys from Ohad's lab can tell you that they do probably genomics all the time. So there's a lot of omics now. There's metabolomics and there's genomics and there's proteomics, and we can actually look at everything and see the whole picture and go by that. Okay, but in general, there is not really a difference, because in many of those methods, what people now show is that they confirm the classical cycles or the classical pathways, and then they add something additional in many cases. So it's really nice to see how those high-end state-of-the-art methods eventually lead or show us or confirm things that were achieved simply by good thinking and a water bath. In general, this is what people have used, right? A water bath and a piece of liver. And this is how these two fields combine. There's no real difference. However, the metabolic communication is able to describe us things that we did not look at before. One example is the fact that the cells in our body can communicate between one another. Okay, so do you have an idea how cells communicate? What is the classical way for cells or organs to communicate with one another?
Hormones?
Come on, I'm sure you know that.
Receptors and ligands.
Right, receptors. So if we're talking about metabolism, which two major receptors you can describe for us? Yeah. So are you and Matan closed on this? Which two major receptors are relevant for metabolism? Come on.
The kinase.
No. Okay, so you're talking about tyrosine kinase. Let's go and be more specific, okay? Which two hormones are highly relevant for regulating metabolic processes?
Insulin and glucagon?
Fine, that's great. Okay, insulin and glucagon. Let me just close you now. I'm unable to see you guys, so maybe you wrote this. I cannot see the chat actually now. It's a little problem here. Okay, so if we have a cell, let's draw a cell. You can see my screen, right? Okay, so if this is, let's say, a liver cell, and what is the major source for energy that comes from outside?
Glucose.
Glucose, great. Here's the glucose. So now we can see that there is a barrier, and this is why we need the communication part, okay? There is a barrier between the actual molecule and the inside of the cell, right? So what brings the molecule in?
Transporter.
Transporter. Do you remember what's the name of this?
GLUT2.
Exactly. This is GLUT, and in the liver, even more specifically, GLUT2. Okay, so these are the GLUT transporters, very important. These are the GLUT transporters, and then the glucose can pass into the cell. Okay, those of you who did your undergrad courses with me probably know those drawings very well. So the glucose can enter the cell, and then there is a whole enzymatic process that begins to form this energy that we will produced from the glucose. So what is the first pathway that the glucose sees for the energy production?
Glycolysis.
Glycolysis, exactly. Okay, so we have glycolysis. So what do we produce during the glycolytic pathway, or in the glycolytic pathway? What are the end products?
ATP.
You remember? ATP. Who said ATP? That's right.
Ani.
Who said Ani? I cannot see you.
Ani Noah.
Okay. Great. I'm looking at the board, so it's very hard for me. And what color is ATP? Everybody knows that ATP is green, right? So here is the first time that we see how a certain molecule eventually provides us with energy, right? So we have the ATP. We have also pyruvate, which is formed, right? And the pyruvate can give us lactate. Okay. And I'm going to ask a big question. I specified here a liver cell, but do only liver cells do that? Can actually take glucose and generate ATP and lactate and pyruvate? No. We know that it's not only the liver cells. Does anybody have another example?
Muscle cells.
Muscle cells, what else? Actually, every cell in our body, okay? But the interesting thing is that it's not only the cells that we usually talk about. There are other cells that we usually don't talk about that can also do that. Only when they do that, we call it fermentation. It's not different, okay? It is fermentation. You oxidize the glucose. So which other cells can do that in our body, which we usually don't refer to? I'll give you a hint. They are found in our gut.
Bacteria?
Microbiota.
Right. The microbiota. I heard Matan, and who else said that? Doesn't know. Okay. So it's the microbiota. They can also do that. So if the microbiota can do the same as we do, okay, they can actually use the same resources, and they can produce all kind of other metabolites that for their own use, for other bacterial use, and also for the use of our body, okay? So how do you think that these cells can maximize the efficiency of the metabolic process? I'll give you an example. Let's say that each and every one of you in the courses that you have exams would study to the exams by themselves, or they will study in a study group, okay? Of course, it's very individual for every person. But my question is, in general, how would it be more efficient?
More ATP.
מה אתם אומרים? זה יותר טוב ללמוד לבד או ללמוד ביחד? אתם יכולים להגיד את זה כל אחד לפי איך שהוא מרגיש.
In a study group.
In a study group. Why, Natalie? Why in a study group? And again, for those of you who want to talk in Hebrew, that's fine. I will translate, that's fine.
Because maybe each person of the group understands better another part and then explains it?
Right. Actually, there is a way to do it even better. If there are, let's say, 5 people who are studying together, 4 of them can go to work and make money, and they will only send one of them to the class. He will pick up all the information. He can come back to the study group and teach everybody, and they will have enough money to buy pizza so that you will have the energy to study even more time, okay? So basically what I'm saying is that when you share resources, it's one way to do it, but in order to share resources correctly, okay, you should communicate, okay? You should be able to know how to send this very person to the class and who is better to go to work and make more money and who is able to has the phone to call the pizza guy to buy the pizza and so on. Okay, so if you share resources and you communicate well, then you are able to do things more efficiently. And basically, maybe you know it or maybe you don't, but the microbiota can actually, the bacteria in your body are able to communicate with the cells in your body, with the immune system, even with the nervous system, through epithelial cells. And this communication actually makes the whole pathways or the whole metabolic process in your body, when it works well, it makes it more efficient, okay? And we will talk about it. And of course, as you said earlier, receptors and transporters and other membrane proteins will be very important in this process. So let's put this cell aside. We will go back to it, okay? But you see that in order to communicate, in order for the bacteria and for the cells to communicate, they must have similar mechanisms to allow them to communicate, right? Because if the bacteria would produce all kinds of substances that we have no receptor or transporter or enzyme that can recognize it, then it would be completely irrelevant, right? So there have to be common communication systems, I would say. It means that if you have an iPhone and the cell has, or the bacteria has, I don't know, a Samsung phone, then you have to use the same network in order to be able to communicate, okay? And as you can see, I gave you one example of glucose and the glycolytic process and all these carboxylic acids that they can actually share and they can use and they can transport between one another. And again, this is very important. So before I go back to these cells, I want to do another zoom out and ask you, the first lesson, you probably learned it in your first lesson in metabolism, and these are the 2 pathways or the 2 major functions that you have in metabolism, okay? Which are the 2 major functions of metabolism? It's obviously catabolism and anabolism. Is there anyone here that can tell me the difference between catabolism and anabolism while I'm drawing? I can wait like 30 seconds until you decide to. And again, it can be in Hebrew, no problem. Wow, that's a big silence.
The use of oxygen and the amount of ATP produced.
תודה, הצלת אותנו. עוד פעם?
The use of oxygen and the amount of ATP produced.
Okay, so now you're talking specifically about what? Catabolism or anabolism?
Catabolism?
Catabolism. Okay, so basically in catabolism, what do? We break down all kinds of substances in order to eventually generate, as you said, I think, okay, correct me if I'm wrong, energy, eventually, right? We need energy. But in this course, you're going to learn something very interesting, that unlike what many of us think, it's not all about energy. Okay, it's really not. We definitely need energy, but in many cases, the evolutionary process does not prefer the, I would say, energy considerations. There are many other considerations in the evolutionary process that prefer other things, okay? And we will talk about it. But now we are, I said, we are in the classical part of our study. So we'll say that, yes, energy is important. I just want to try to change the color here. Come on. Okay, so we produce ATP. And what else? What other molecules are very important for the energy production? Come on, all those of you.
NADPH.
Great. Oh, yeah. I'm just telling you, Luis is listening. So all those of you from the MD class, make sure you say the right things. Yeah. So what are those? NADPH, NADH.
כוח מחזר.
Yeah. Diffusing agent, diffusing power. Let's write it this way. Very, very important molecule that can contribute. Oh, I forgot the D. Excuse me. Okay, so this is what happens in the catabolic processes, right? We're using all these different substances. We can also use oxygen, of course. Okay, and all kinds of carboxylic skeletons. And other animals can actually use light as a source of energy as well, but we're not going to do this because this is not life sciences. Although we do have Amit here. Amit, you can always contribute to us with your knowledge in bacteria, plants, or what have you. Sharks. We will need it. Okay. What else? What are we producing in the catabolic process? So you said ATP, and this is all the good stuff. We break down all the carboxylic stuff and oxygen. What are we getting?
CO2.
That's great. CO2, obviously. What else? When we break down proteins, wow, I didn't notice that I drew it so big. When we break down proteins, what do we get?
Nucleotide.
Mamit?
Nucleotide.
Nucleotides, okay.
Amino acids.
It's relevant. What, Natalie?
Amino acids.
Eventually, the amino acid would break down and we'll get the ammonia or the ammonium. Okay, and this is what all creatures do. Eventually, we end up with CO2, ammonium, and the third major thing, which we like a lot, water. Okay, and then when we do the anabolic process, we're actually using all these. Okay, we're actually using all these. We're using the ATP, we're using the reducing agents and so on. And what do we get? First of all, obviously ADP, okay, and NAD, blah, 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 plus, okay. Of course, it's either NAD plus or NADP plus. Okay, there are of course a few others, but these are the major molecules. Okay, what are we using them for? In the anabolic process. So in catabolism, it's pretty obvious. We take all these carboxylic acids, we take other creatures use light or the photon energy, oxygen, and so on. But eventually, in the anabolic process, what do we do with all that? With all the ATP we generated? Think about it. I'll give you 1 minute because it's important and it's not something that we usually think about.
To absorb phosphate?
Again, Matan?
To absorb phosphate.
To
Phosphate
absorb
regulation.
phosphate regulation. So saying basically to do some kind of work, right? Phosphate regulation, absorption, and other things are definitely relevant, okay? But what we use it for is to do some kind of a cellular work because we can use the ATP and soon we'll see it, how we even need to use like a battery in your cell phone. We need to use the ATP just for the communication. It means that we spend energy in order to maintain communication between cells, metabolic communication. We'll see how, okay? But in general, we want the cells first to grow. We maintain the cells, we maintain the living, right? Using this energy of ATP and the other metabolic stuff. And we also do functional and mechanical work. For example, we need our muscles to contract. To do that, we need ATP. We need our kidneys to start pumping the water and the solutes. And Matan gave us an example, okay? In order to do that, we need energy, okay? So, and obviously also to build the cells, to add more cell mass. Okay, so for cells to divide, for a tissue to regrow, okay, to regenerate, okay, and Aaron, are you still here?
Yes.
Great, okay, so we're talking about tissue regeneration here. So for the tissue to regenerate, we definitely, for wounds to heal, we need all this energy, right? So all these processes that we've just described, and in general, I can say work, okay, different, we described all kinds of work that the cells can do, osmotic work, cellular work, mechanical work, and of course, for adding the biomass or growing the biomass. Okay, we need to do all that stuff, okay? So what we have described here are the very basic concepts of metabolism. And it's very general because all creatures actually utilize these processes in different ways that they have developed in different pathways. But in many cases, there are many similarities between them. And we gave an example of the bacteria versus cells, okay, human cells or mammalian cells. So in general, they are capable, they have all the machinery that can allow them to communicate with one another and to share resources, okay? And this is why metabolic communication is so important, because when we evolved from the bacterial mass to the complex organisms that we are now, this had to occur, okay? These mechanisms had to become highly efficient and to work together and to share resources, okay, in order to form higher organisms, okay? Any question thus far? Or anything you want to share with us? Any insights? None. Okay. So I want to go back to this cell here that I drew. And as I said, we are now doing an overview of metabolism and communication in general. And I want to ask, why do we even need, okay, to change these processes? Why are there changes in these processes? Why do sometimes we need one pathway to function and sometimes we need another pathway to function? What changes during our lifetime that requires those changes? Why can't we just have, be like a bacteria, and even they don't do that, sit in a very, very defined environment and just live there and reproduce and, just keep on living this way? That's a little philosophic, I know, but you'll have to excuse me. What changes? What changes during your life every day that requires those different adaptations? Don't even tell me the whole story. Just give me one example of something that requires a metabolic change in your life.
I think the substance that we have, like if we just ate or we are after a fast or something like that, and also
Wonderful.
the activities we try to do. If now we want to rest or we want to run, then we need different processes.
Wonderful. Thanks, Ofek. Thank you for this contribution. This is exactly it, okay? As Ofek said, we don't do much. We either eat or don't eat or run. For those of you who still run, or move, let's say move, let's not exaggerate. Okay, that's exactly what we do. So the biggest changes that we have in our life is either we are fed, we're eating, and now the entire body has to adjust to the high amount of potential energy that it gets and distribute it correctly. And that's what it does in order to prepare us for the times where this food is not going to be available. Okay, and that's fasting. And of course, that if you have another risk, or you always give the fight or flight example that I'm sure you all have heard about, is the movement, the major changes in movement. Like, for example, what we do today is exercising. So when we exercise, we have to change these entire metabolic processes to allow us to change between anabolism and catabolism, and it has to occur very fast, okay? And in order for us as a whole organism, for the different organs in our body, for the different tissues in our body, for the different cells in our body to adjust to this changing condition that has to occur very fast, obviously, these cells have to talk to one another. They have to describe to one another the changing condition, adjust to it as fast as possible, okay? So the communication is very important for the adaptation to changes in energy, okay? And the metabolic product demand actually changes as well. So they all have to prepare for that. So I think that now we are starting to understand that there is an importance not only for the metabolic processes to occur, but even more so to the, I would say, to the communication, simply the communication between those different cells, okay? So now let's go back to this cell that we talked about and zoom into it again. We described the glycolysis, but where do we get most of our energy? In most of our cells. Where does it happen?
Krebs cycle.
And where is the Krebs cycle? Where does it occur within the cell?
In the mitochondria.
In the mitochondria. Okay. Just a second. Oh. That was Aaron. Aaron, you shared with us? I don't always see who.
Yes, yes.
Okay, thanks, Aaron. Okay, so let's draw the mitochondria. And what do they say about the mitochondria? Where did it originate from? How did we get mitochondria? What is it actually? Going back to what we've discussed earlier about sharing between different organisms. So what is the mitochondria? What is it actually? What was it in the past?
A bacteria?
It was definitely a bacteria. Well, I wouldn't say definitely, but this is what people think, that it was a bacteria because it's very similar to a very specific type of bacterium that we know. Amit, do you remember the name of the bacteria that is similar to the mitochondria? If I'm not mistaken, alpha protobacterium. I think it was this one that now also exists, and it's highly similar in many, many aspects to the mitochondria. It obviously has the inner and outer membranes. And as Aaron said, now the pyruvate can go into the mitochondria. And then it goes into a specific pathway that is called the Krebs cycle. Does anybody know another name for the Krebs cycle in English?
TCA cycle.
Right. Who said that?
Amit.
Amit. Thank you. That's quite right, and it's much easier for me than writing Krebs. So it's TCA cycle. Do you know what's the acronym for? You remember? Okay, it's for tricarboxylic acid cycle, okay? Because mainly all that we've talked about, most of the molecules that we talk about here that are important for the metabolic processes, many of them are carboxylic acids, okay? Basically, all the TCA cycle intermediates are carboxylic acids, okay? And what do we produce in the TCA cycle?
Carbon dioxide and ATP.
Okay, so if we go back, let's go back to what I wrote before. You see the catabolic and the anabolic processes. So this is definitely a catabolic process, since we can use all these carboxylic acids and we can generate ATP and also CO2 and also reducing agents, or reducing power. Okay, so we make all this CO2 and other stuff, but the most important thing, of course, is and usually it's a big secret in the introductory courses in the undergrad studies. What does the NADH do? Because everybody knows about ATP. But since you guys are already postgraduate, then you already know what the NADH does. What does it do?
It's used in beta oxidation.
Who said beta oxidation? Wait. Who said beta oxidation? Come on, just Nadav said beta oxidation. Okay, so do we utilize NADH in beta oxidation? You're asking Matan. Okay, Matan, do you have a better answer?
I think, and don't really remember the pentose pathway.
That's fine. I hope Luis is listening. Did you study with Luis?
שרת מעבר האלקטרונים.
Nice. See, this is why biology. Okay, wonderful. Toda, Amit. Okay, I think you should You should sit with those two guys in your lab and, teach them one or two things. It's very important. It's a good thing they have you. Okay. Do you guys remember oxidative phosphorylation? Okay, very important oxidative phosphorylation. Ruri, are you with us so far? Are those things completely new to you, or you are familiar with them?
No, I'm familiar with it.
Okay, great. I'm happy. Great. Okay, so
Sorry, I'm shy.
Sorry.
I'm really shy, so I'm not really like interacting.
That's completely fine. Just feel free to learn any way you want. Okay, whenever you feel you want to swoop in, that's fine. Okay, we'll be happy to hear what you have to say. So eventually, in this entire process, we generate the reducing power, of course, and what I'm drawing here is, of course, the ATPase, and we can generate using this reducing power, the reducing agent. They move into the phosphorylative oxidative phosphorylation, I'm sorry, okay? And there is a whole process for those of you who want to go back to Leninger and try to remember it. So you generate a proton, a very huge proton electrochemical gradient that eventually, by moving electrons through the entire, through those complexes of the oxidative phosphorylation pathway, you remember there's 1, 2, 3, and 4, and then when the proton gradient is formed, okay, it enables the ATPase, which is the orange machine, to generate ATP, and then this ATP can go and be utilized later as energy, as an energy source. And now, since we know that Matan, wait a minute, that Matan and Nadav, right? Matan and Nadav, they have issue with fatty acids. So we'll remind them that from the TCA cycle, during when we're shifting to an anabolic state, we don't use the carboxylic acids in order to form energy. What we can do is that we can take the citrate out of the cell, and the citrate can be utilized to generate fatty acids, right? But those fatty acids in a different pathway, when it's the catabolic pathway, the fatty acids can come from the outside, okay? Who sends fatty acids to the cells? Where do they come from? What is this?
Apoprotein?
Well, this is how it's usually packed in the liver and sent to the tissues, okay? But when the tissues or when the liver wants to utilize fatty acids in a process called beta oxidation and does not require the use of NADH, but rather the formation of NADH or the production of NADH, then usually adipose tissue, adipose cells, they send out the free fatty acids to the bloodstream, and then those free fatty acids can go into those cells, to the different cells. Of course, muscle can use them as well, right? You remember muscle really likes fats, and it goes into the mitochondria eventually, and I'm not going to go through the entire process of the CAT1 and CAT2 and things that you all probably don't remember from your undergrad, but then it goes into the beta oxidation pathway. And in the beta oxidation pathway, we also generate a lot of NADH, much more from fats than we get from sugars, and they can also be utilized in order to generate energy through the oxidative phosphorylation pathway. Okay? There is maybe one last process that I did not describe. Can anybody think? It's an anabolic process. What is missing from the picture? Let me start here. I'll just show you that pyruvate can go into the Krebs cycle and make a bypass. Okay, I did not mention a very important molecule here, of course. In the middle, that from pyruvate through PDH, we get acetyl-CoA, which is very important for the energy. Maybe I should draw it better. While you're thinking about the process that I have left out, come on.
GNG.
Great. Who said that?
Aaron.
Yeah. Okay. So this is acetyl-CoA, and we can bypass. Okay, this is the first. Acetyl-CoA is obviously the first metabolite that is generated to the TCA cycle from pyruvate through the PDH, of course, and it's also generated. I mentioned NADH, but we also have FADH2 and acetyl-CoA that are generated in the beta oxidation pathway. Okay, and can be used for everything, but those of you remember your regulatory processes probably remember that during beta oxidation, the high NADH and acetyl-CoA actually inhibit the TCA cycle at a certain point. And of course, now I'm going back that pyruvate through pyruvate carboxylase can bypass the acetyl-CoA and the TCA cycle to oxaloacetate. I will not go into it. And we can actually start a process in which we reverse glycolysis and we bypass the major regulatory enzymes until we generate glucose. Okay? Any questions about this very clear drawing that I just drew to you? Or as I call it, glycolysis metabolism in a nutshell. Come on, I want to know what I want to understand what you guys are thinking. That was this helpful in reminding you things, or are you now completely lost?
It is helpful and reminding. I think it's been a long time since I've let heard all of these cycles, and it's way to hear it all together.
Okay, that was Ofek, I guess, right?
Yes.
Okay. Where are you now, Ofek? Are you already in Haifa?
I'm almost at home, almost. Okay. And I have Loki here with me, so now there's one more.
Okay, wonderful. Okay, the marvels of Zoom. Talking about Zoom, now I've zoomed out, and you see this entire stuff that we have talked about so far. And now the question is whether you want a break, because I think I have something like 30, 40 minutes talk, maybe even less. Would you prefer a break of 10 minutes, or do you want to simply continue? Let's vote on it. All those of you who want a break, raise your hand. Who wants a break? Raise your hand. Natalie, I'm afraid. Yeah, the majority has spoken. We're going to continue and finish around 4, okay? For all those of you who will not fall asleep by then. Okay. let's see what else I wanted to talk to you about. So basically we started talking about these processes, and you can see that for example here, the adipose tissue generates free fatty acids that can be utilized by, let me draw this more clearly, by the liver, for example, liver cells. Now, liver cells, under, as we said, changing conditions, right? When do we use the free fatty acids? When we said that we are talking about adaptation to change, changes in the energy and metabolic demand. So under which conditions do we utilize free fatty acids?
After you use glycolysis and after you use the Krebs cycle, like
okay.
When you run for 30 minutes.
Okay, and what about when we are fasting, for example? Is it the same?
Yes.
Okay, and of course, what would define that, which processes will take place, whether they're catabolic or anabolic, in response to the changing environmental changes, I would say, okay? In many cases, these are hormones, right? So when we are talking about the very basic type of communication between cells that allows us to sense the environment and respond to those changes in metabolic demand, it's basically hormones, right? And more specifically, we can talk about the classical ones, which are glucagon and insulin, right? So when free fatty acids are released from the adipose tissue and utilized by the liver, how does the adipose tissue know that it has to release free fatty acid? How does the liver know that free fatty acids can now, or not only the liver, but also the muscle, that they can actually use it now, okay? Which two hormones are the major ones under these conditions? Do you remember? So when we are fasting, it's glucagon, right? So in order to sense that, the changes in glucagon, all these cells have the glucagon receptor. And the glucagon receptor actually is the signal for those cells telling them which metabolic processes have to be activated and which metabolic processes have to be inhibited. Okay, that's how it works. And it's always this ratio between the glucagon receptor and which other receptor?
Insulin.
Somebody said anything?
Insulin receptor.
Right, the insulin receptor, which are completely different, than one another, okay? So now we understand something important, okay? In order for these environmental changes to affect the cells have to know what is going on. And in order to understand what is going on, some signal has to come and tell them what are those changes. So if we are talking about fasting, tzom, it's glucagon. When we're talking about feeding or eating, okay, that we have a lot of energy coming, then it's insulin. What about exercise? Which hormone is in?
Insulin for
Sorry.
if you are insulin, if you are like doing exercising, you need insulin.
Insulin, and what other important one? There's another important one that usually affects the muscle. It's adrenaline or noradrenaline or epinephrine or norepinephrine. Okay, so these are very important also during exercise, and they will definitely affect and shift the metabolic state and also the metabolic pathways. They will activate those that are required. They will inhibit those that are not required under these conditions. And there are, of course, and this is the entire undergrad introductory class or course discusses those different regulations and how they occur. Okay, and of course, we're not going to get into it, but only those that we will actually need. So what we've described now with glucagon, insulin, adrenaline, these are basically the most important, sorry, not the most important, the classical and most basic regulatory functions. They are highly important. It's not something that we can disregard, but we now know that there are many others, novel, more complicated types of regulations. And this is what we are going to discuss in this course, because basically the microbiota, at least as far as I know, okay, they do not produce glucagon or insulin, okay? But they do produce short-chain fatty acids. They do produce carboxylic acids of all kinds, okay? And these are used for the communication between them and between our cells, the host cells, the epithelium, the muscle cells, the immune system cells. And we're going to talk about this a lot, about all those new regulatory pathway of communication between those different cells. Okay, so we've described the adipose tissue, we've described the muscle, we have the liver. When we produce glucose in the G&G process, is it a catabolic or an anabolic process?
Anabolic.
It's obviously an anabolic process. What do we need it for? I mean, the liver, it uses energy. If we had liver cells in a culture, just growing and living in a pond or something, they wouldn't have to do that because they would lose glucose, they would lose energy. So there is a reason why they decide to give up this energy, give up resources, which usually creatures don't do. Okay, let's look at the liver cells as creatures in their own right for a second and think whether these creatures, why do they do that? What is the incentive for a specific cell to give up energy? Okay, in this case, the classical notion, okay, and you probably talked about it a lot. Why does it produce all this glucose? Who needs it?
The body.
Yeah, but specifically, who in the body requires this glucose?
The brain.
The brain. Great. Who was it? Nadav?
Matan.
Oh.
No, it was Nadav.
Matan or Nadav? Come on, who was it? Because I
Nadav.
Looks like Nadav is, his biochemistry is starting to come back, right? Yeah, okay, that's good enough. So, yeah, the brain, definitely. There it is. Why does the liver need to give up resources for the brain? And I'm not talking philosophically. It's not philosophically speaking. Why does the brain need glucose and cannot use other stuff or produce its own glucose?
Because it needs a regulated and constant amount of glucose, of energy at all times, not dependent on the state of the body.
Mm-hmm. Okay, so basically in general, the brain is not equipped, okay, doesn't have the pathways, doesn't have the enzymes to use fat, for example, okay? It has to use either glucose that is produced for it during fasting by the liver, right? If it doesn't come from the outside, it has to come from somewhere, otherwise the brain would stop functioning, okay? And of course, there is another molecule that the liver produces for the brain. Do you remember what it is?
Ketonium?
Yeah, great. One, ketone bodies, okay? So the brain and also the other organs in the body can use either glucose or ketone bodies. Pardon my writing. That's all I have, okay? So we can see something very interesting occurring, okay? Even though the liver cells are in their own right, they function all the time, they live and they want to keep living like any other organism, they are willing, and that's why I said not everything we have to take into consideration energy considerations, okay? Not everything is energy, because apparently a cell is willing to give up resources, energy resources, like glucose, like ketone bodies, and it, under certain circumstances, even uses energy in order to do that. It loses so much just in order to help another organ or another cell, okay? But if you look at it, and now we're talking only the classical metabolic processes, you will see that all these arrows, what do they indicate? I'm talking about the arrows that go from one organ to the other.
Metabolic connection?
Right. There is a metabolic connection. There is a metabolic communication between these organs all the time. All the time. Okay? And this is not just communication. It's not just the communication. Because if it had it been only communication, then the cell would not have given up much, okay? It's more than that. They share resources. They share energy. They share molecules that have energy potential, okay? It means that there is some kind of, I don't know, some kind of a big idea behind this, that they are willing to give up the most important thing an organism has, okay? And as a matter of fact, we don't see it only in higher organisms like humans and mammals, and we also see it between bacteria, okay? They are willing to give up the most important and precious things they have because they gain something from it, okay? They gain something from it. They would gain protection, they would gain survival in some kind of way, and this is why they require a constant communication between them, okay? And this is just in the, on one level, okay? We say that because it's some kind of a nice idea, but there is another major question that comes up, and this is, how do they do that, okay? And I think this is the most important question that we're going to answer in this course. How can, not just the why, because the why would come up and it's not really a scientific question. We can understand it when we learn the mechanisms as we go. But the more important question is here, how? How can they do that? How can a bacteria talk to an epithelial cell of a human, okay? And they would do that in order to share resources. And it's probably because they both benefit from it, okay? So this will be one aspect of the communication that we will discuss. And another aspect that is going to be also very important for us is to understand what do they benefit from it, okay? What is it exactly that these cells benefit from in order to do that? And the third aspect that we will talk about is what happens when this communication goes wrong, okay? In one word, what do you think would happen when a communication between cells in our body goes wrong.
Both sides loses?
Both sides lose, not necessarily. You will see that it's not necessarily that both sides lose, okay? But let's think of ourselves for a second. What would happen to us if we lose? In one word. 10, 9, 8, 7. I'm waiting.
But we will die.
What?
We could die.
We could die. Okay. So you took it one step ahead. I was thinking more of a disease or a pathology, but I guess that's how MDs think. There is no way to save the patient, right? Okay. Yeah. So disease, and as Ofik says, death. Okay. So yeah, that's how it's going to affect us. That's, as you probably understand, is not a good thing. Okay, so in this whole picture that I put here, we can add all kinds of other organs, because basically all organs are involved and they talk to each other all the time. Okay, so we've mentioned the microbiota, right? So of course, the gut would be very important in this mechanism, and the microbiota in it. And when we're talking about catabolism and anabolism and so on, then we know that through the air system, we will bring all the O2 into our body. Through the upper gut system, we take food into our body. And we always thought that the food that we are eating is eventually ending up somewhere in our body, okay? We thought, okay, if we're eating an apple, or yeah, let's take an apple, for example. There's a lot of glucose in the apple. So we thought, okay, let's see how much glucose we have in the apple, okay? It goes into our body. Here is the apple. Okay, so we have all this glucose that went into our body from the apple. And pardon my drawing, we will see how much of that came out. And then we'll know how much glucose we absorbed and is now moving in the circulation, maybe to the liver and maybe to other places. Okay, but now, when the microbiota is suddenly important in all these metabolic processes, we understand that it's not always the case, okay? In many cases, we can eat an apple with a lot of glucose and get suddenly fatty acids, okay? One example, and this is from studies that come from the Weizmann Institute of the Microbiota, is that all the people that want to lose weight and they are using all those sweeteners, right? Like, I don't know, sucralose and all the other stuff, sweet and low, for those of you who know these sweeteners. We now know that in some people, the microbiota content in their gut can take up those sweeteners and generate, do you know what? Any ideas?
Fatty acids?
Fatty acids is nice, but what would be the worst thing that you can think of for people who are using sweeteners?
Glucose.
Exactly. Okay, there are people that their microbiota content is such that the bacteria there know how to take specific sweeteners and turn them into glucose. And then they don't understand why they don't lose weight or why their blood glucose levels are still high. and that is the reason. And there are other fortunate people that when they eat bread, for example, and we say, okay, it's white bread and all these empty calories and so on, but it turns out that their microbiota can take the glucose and other substances in the white bread and generate fibers, okay, which are very good for you. And then suddenly you understand why other people consume so much white bread and carbohydrates, but somehow they don't gain weight, okay? So this is how important the microbiota has become. And those changes that we are now experiencing due to the understanding that microbiota does other things. Okay, and now I'm going to move to the very last part. I promised you 30 minutes. It means that we have about 10 or 15 to go. Oops, that's not a good thing. Okay. And now I want to discuss or start funneling into the communication processes, okay? And what mechanisms we have in order to generate this communication between different organs, maybe also between microbiota and our own cells, and between the immune cells and epithelia, and you just name it. Okay, so I will draw. I don't know. I don't want to use the other, already too packed. So obviously, which first proteins are going to be the ones that encounter the extracellular substances that arrive? Let's say that the microbiota generates, I don't know, huge amounts of short chain fatty acids. Just an example. And we'll talk about, I'll just tell you that short chain fatty acids they have the characteristics of fatty acids, okay? But their chain is very short. They usually have 2, 3, 4 carbons, not more than that, okay? And we're usually talking about 3 major types. The first is acetate that has 2. The second is propionate, if I'm not mistaken, that has 3 carbons. And the third is butyrate that has 4. And these, for example, I'll just throw some ideas, but we know today that short chain fatty acids that are produced by the microbiota from consuming fibers, dietary fibers, they're very healthy for you. For example, they have anti-inflammatory effects, major anti-inflammatory effects. That's why they offer people who suffer from all kinds of inflammatory disease to eat more fibers. Okay, in this course, we will study that it's not always the case, okay? But let's say that a cell suddenly, okay, encounters the short chain fatty acids. Let's take, for example, a macrophage, okay? A macrophage residing in the gut, okay? MD people and all others, where are the macrophages found in the gut?
In the mouth?
Where? Where? Are you asking?
In the mouth or the mucosa?
Okay, let's say that they are found in the submucosal layer, okay, under the epithelial layer. Okay, in the submucosal layer, we have many macrophages. These are usually monocytes that travel to the tissue, and when they infiltrate the tissue, you can find them in the submucosal layer in the gut. Okay, so let's say that the bacteria produced a lot of short chain fatty acids, all those that I mentioned earlier, and they are being absorbed through the epithelial cells. And now the macrophage, which I drew here, This is a macrophage for those of you who don't know. Now the macrophage sees the short chain fatty acids. What can it do with the short chain fatty acids? I mean, at the very first moment that it sees the short chain fatty acid, the short chain fatty acid approaches the cell. What do you think would happen now? Does it ignore it, or can it ignore it?
It should ignore it, no?
It should, you think? Why? If you say something, then just explain why.
Maybe because there will be an immune response for short chain fatty acids.
An immune response for short chain fatty acids. Who said that? I'm sorry, I didn't look at the screen.
Right. Matan.
Okay, so Matan says that's fine. You know, there's no right or wrong here. I usually don't ask a yes or no question. So Matan says if a macrophage sees short chain fatty acids, I'm thinking short chain fatty acid might affect it, and therefore it should ignore its existence. Okay, so if had you been right, it means that there are no other cells that would take short chain fatty acids into consideration or use it. Okay, they would simply ignore their environment all the time. Is that true? Do you ignore your environment?
No.
No. So basically, I would say that Matan is right, but is also wrong. Okay. So how can you be right and wrong in the same time? My explanation is that if the cell has some kind of mechanism that, you know, has eyes or hands that can see the short chain fatty acids, and you're right, not all cells can see it. Okay. Not all cells can know that there are short chain fatty acids in its environment. Then you would have been wrong. But if there is no I don't know what I said. If I said that there is a mechanism, then you are wrong. If I said that there isn't a mechanism, then you are right, okay? So it's all about what the cell is equipped with, okay? Which equipment does the cell have to carry with it in order to sense short chain fatty acids? Come on, people. Now I'm looking at the other side of the screen. I see Natalie falling asleep. I see Bayan looking at me. I see Noah now. I see Amit. Come on. Amit, I will ask you directly. Who said receptor? Who helped Amit now?
Matan.
Matan. At least be nice and you can whisper it to her without all of us hearing, right?
I knew the answer.
הוא אומר נכון, אבל אני בטוח שעמית ידע. I'm sure Amit knew it. Sure. Okay, so sure, a receptor. What is a receptor? Now Amit will tell us.
חלבון שהוא יש לו ליגן שמתחבר אליו, ובהתאם לזה הוא משפל תגובה תאית.
Great. So what Amit basically says is that a receptor is some kind of protein that is embedded within the membrane, okay? And it faces the extracellular environment, and it also faces the intracellular environment. And this property of this type of protein allows it first to sense specific molecules. So obviously you all know that not all that receptors are very specific, okay? They have a they are equipped to do or to sense very specific molecules. It means that in order for short chain fatty acids to be sensed by the cell must have a receptor which specifically knows how to bind short chain fatty acids. And if it won't have this receptor, then Matan is completely correct. It wouldn't even know that there are short-chain fatty acids in its environment. But if it has these receptors in its membrane, then it would know that there are short-chain fatty acids. And now it's a completely different story. Because let's say that the bacteria knows how to sense and generate short-chain fatty acids, and the macrophage knows how to sense and utilize short-chain fatty acids, then we formed a communication pathway between them. And that's, maybe it's simple to say, but this is something huge. It's something fascinating that completely different organisms can actually communicate with one another using the same equipment, okay? And it's really, really fascinating. And these are the things that we are going to talk about a lot in this course. Now, it's a nice thing that the short-chain fatty acid can be bound by the receptor, okay? It's very nice. But what does it actually do? And Amit says that it propagates some kind of a pathway within the cell, okay? Or some kind of signaling mechanism that would eventually lead to some kind of final effect, okay? And soon we will describe, and by this I'm going to finish, a series of the major receptors that we know. Okay, so this is one aspect. Maybe we'll do it at the beginning of next class because I see you guys are getting tired, and that's fine. So we know now that certain cells can sense the short-chain fatty acids, and of course it's relevant to other substances as well. But I said something else. I said that it can either sense it or use it, or both. So sensing is obvious, but can a receptor use the short-chain fatty acids? Or can a receptor allow the cell to use the short-chain fatty acids, the actual molecule?
It might.
How?
Maybe it can enable internalization of the receptor and the fatty acid to the cytoplasm or something like that.
That's very interesting what you said, okay? And this is something that I did not think about even, okay? But my question is, usually each receptor binds a single molecule of short-chain fatty acid. So you would have to produce, I don't know, a 1-to-1 ratio, stoichiometric ratio of receptors to short-chain fatty acids in order to do it properly. That means you would, in order to get a very small amount of short-chain fatty acid, you will use huge energy to produce and to send all these receptors to the membrane. It's highly inefficient to do that. But having said that, do you know of proteins that can allow the flux of short-chain fatty acids into the cell?
Maybe kind of a channel.
Great, Amit. Thank you. Okay. Do you have a general name for channels and others?
טרנספורטר.
Great. תודה. מי אמר טרנספורטר?
אהרון.
Aaron. נהדר. Great. Okay. So in order to take the short-chain fatty acids inside, it would be much smarter to use a transporter of some sort. And we know that transporters are also specialized in transporting or in facilitating the flux of very specific molecules. And in this case, it would be a short-chain fatty acid specific transporter. And in this way, we can sense, or the cell can sense, the short-chain fatty acids in its environment. And if it also has the receptor, then it can also take the short chain fatty acids into the cell. And now it's a completely different story. It's a completely different story. And these two major types of proteins are maybe the major communication gear that a cell has in order to generate, produce, secrete, absorb, and sense different molecules. And it has to be highly specific, but not only that, it can use the system in order to even transfer signals, metabolic signals, to other cells. So now think how complex it is. It means that if bacteria talks to the macrophage in some sort, somehow, and they do, okay, and we will have specific manuscripts and the papers that we will focus on and discuss and show how these things are done, they can actually do all these things. They can exchange those molecules between them, but it's not like hormones. It's not like glucagon and insulin that do nothing except then sending the signal, okay? They can use them for signaling, but they can use the same substance, the same molecule for energy and for all kind of other metabolic processes that the cell requires, okay? So now I can see that you guys are getting too tired, and that's fine because we talked about many things today and we saw a very big picture. So I will end here. Next time, I will go a little bit more. I will think, okay, I will be in touch with you. And I think that, that I will talk to you a little bit more about those communication methods. Okay, maybe at the beginning of the course, at the beginning of the class, I'm sorry. And I will also update you on the website as to papers that we will start working and discussing, as I mentioned in the beginning. Okay, so the first time you're going to do that, we're going to divide into, we're going to use those, what do they call it, breakout rooms, in order to divide to, I think, 2 major groups, where you will discuss things. And as I said, in each class, we will focus on one of your scientific research topics. Okay, so please go into the Moodle. I will update it today and put your topics inside that, so that we can start arranging those things and combining a specific journal to your topic, and that's it. Okay, that's really it. Do you have any other questions before we say goodbye? No. Okay, so thank you very much. It was a lot of fun talking to you and meeting you, and I hope we'll see each other next week, right?
Thank you.
Thank you very much.
Thank you.
It was nice. Bye-bye.
Bye bye.
Thank you. Bye-bye.